Hello, I'm Scott Burks at the NIH Clinical Center, and today's presentation is going to be discussing transcriptome-wide changes in murine melanomas and breast tumor models using different pulse-focused ultrasound exposure times. So the objectives of this are really two-pronged. We want to look and see how different tumor types respond to what would be identical PFUS exposures, but also in pulsed ultrasound, often the number of pulses a tissue receives seems a bit arbitrary, and so we really wanted to look if the responses were further altered as a function of the number of pulses the tumors received. All right, so to accomplish this, we used 4T1 as breast tumors or B16F10 melanomas. Mice were bilaterally implanted and tumors were grown to about 5 millimeters. We then sonicated them. Every tumor got these parameters where they had treated the entire volume using 2 by 2 millimeter spacing. However, we did break down into different treatment groups where the tumors received either 10 seconds or 100 pulses, 20 seconds or 200, or 50 seconds or 500 pulses. And then each of those groups was euthanized at 2, 24, and 72 hours post-PFUS. And we did bulk RNA-seq with pretty standard analysis like GSEA, KEGG, and gene ontology. We obviously get back an enormous amount of data, and we started with really what would be a 30,000-foot view where we looked for gene ontology terms that described biological processes and whether those were enriched or not due to pulse-focused ultrasound. Now, for example here, this is the 20-second exposure at 24 hours post-PFUS, and we see the 4T1 and the B16s actually respond very differently. So, for example, cell motility is increased in the 4T1s, but metabolism terms are all downregulated, as well as cell cycle terms are downregulated. If we look at the B16, the gene ontology terms for sort of generic cell signaling are all down, but interestingly, we do get reduced extracellular matrix signaling. But importantly, gene ontology terms for immune function are dramatically upregulated. So if we hone down on these general biological processes with slightly more focused analyses, what we see is if we run the 4T1 data through a metabolomics gene set, we see very specifically downregulations in glucose metabolism, as well as upregulations in fatty acid and protein metabolism. And if we look at immunogenic genes in the B16, we see activation of largely innate immunity, but even some small activation of adaptive immunity gene sets. And if we compare how these change as a function of pulse number, so for example, this is 500 pulses versus 100 pulses, we see in the 4T1 there are a couple of dozen things about glucose metabolism that are further downregulated by giving the additional pulses. However, it's a little different on the B16. These are a few nodes that had to do with cytokine signaling, but largely the immune function was not further upregulated by giving the additional pulses.
So if we look at heat maps for individual pathways here, and I show two as an example, we see that they fall into two primary categories. First is that there just isn't a lot of overlap as far as what is expressed by the two tumor types, where we see a large divergence. Some genes will be expressed by the 4T1 and not at all by the B16, and vice versa. The other type of heat map that we see is where there are a lot of common genes being expressed, but if you look at expression patterns, they are different between the two tumors. So for example here in this metabolic pathway, if you look at the last two columns on the right for the 4T1, we see that there is quite a bit of suppression from baseline, whereas if you look at the exact same genes in the B16 following sonication, there is substantial upregulation of those genes. So the last thing I want to show is how do these transcriptomic changes correlate with tumor growth in an animal or inhibition of tumor growth by pulse-focused ultrasound. So what we're looking at here are tumor volumes 5 days after pulse-focused ultrasound for both the 4T1 and the B16. And what we have repeatedly shown previously is that the conventional 10-second ultrasound condition yielded a significant inhibition in growth rate for both tumor types. And we see in this experiment, we are able to replicate those data. However, if we look at further changes in growth from the 50-second ultrasound condition, what we see is that for the 4T1s, there is no additional inhibition of tumor growth, in spite of the fact that a lot of the genetic changes occurring in the 10-second pulse-focused ultrasound group, the magnitudes of those changes are even greater in the 50-second group. Yet it doesn't seem to pay much dividends as far as inhibiting tumor growth. If we look at the B16, however, it is a different scenario where, again, we see magnitudes of gene changes are greater with the 50-second pulse-focused ultrasound compared to the 10-second. And here we actually see that it's correlated with a greater reduction in tumor growth rate. So what we want to look at now is, we're hypothesizing that maybe the gene changes for the 4T1, you reach some, adequate expression level, but further increasing them, doesn't do much as far as further inhibiting the tumor growth rate, whereas the genetic changes here seem to be more tunable, that for the B16, where you get some reduction in tumor growth rate with some activation, and that trend continues, that you get more reduction in growth rate with additional activation. So those are the kinds of things we're going to look at going forward from here. All right, so to wrap up here, it is clear that giving identical sonication parameters and protocols to different tumor types generates very different tumor-specific responses. And while that general statement being true is somewhat predictable, what we're seeing here is that these additional pulses can both amplify certain genetic changes but does not affect others. And when we look at the effects on how tumor growth occurs in the animal and whether it's inhibited, we see again different tumor-specific responses to the same treatment protocols. And so what we want to do from here on out is to start looking more into physiological studies, as all the genetics are really correlative at this point. We want to look at things like molecular imaging, further looking at immune function studies, metabolic studies, and those kinds of things, with the goal of trying to identify what is the biological characteristic or characteristics about a specific tumor that would make it a good candidate for PFUS treatment. And further, we really want to understand how these different sonication parameters generate very specific biological changes. And what that hopefully will allow us to do is then to match biological characteristics of tumors with a specific PFUS protocol that would make best use and most efficient use of PFUS as a tumor therapeutic. So with that, thank you very much for listening. I again acknowledge everybody in the Frank Lab as well as NIBIB for support. And if you have any questions, please don't hesitate to email me at scott.burks@nih.gov. Thanks again for listening.